talked about how we would go about catabolizing the carbohydrates that would be in the slices of tomato. However, that was not the only thing in that lovely caprese salad. There was olive oil, so lipids, fats. There was ricotta cheese, more lipids and fats. So we need to consider that for our microbes, it's the same way. That is, carbohydrates, sugars, are not the only things on the menu. Also, as humans, we are evolutionarily, genetically wired to crave fats. And what we're going to find is that that's because fats are very nutritionally dense. That is, we can get even more molecules of ATP from catabolizing a fat than we can a sugar. So that's why we have sayings such as living on the fat of the land, or killing the fatted calf to welcome someone home. It's important to note that for much of our history, we humans had trouble finding enough to eat, getting enough calories. And one of, again, the most energy dense foods that we could get would be fats, lipids. And so our figure to the right shows the basic pathway, an overview of it, for catabolizing lipids, notably fats. Other lipids go through slightly different pathways, but we're going to talk about now fat catabolism. So, as shown on the previous slide, and now in more detail on the figure of our current slide, what we have is at the top of our figure a triglyceride of fat, and that's going to be absorbed into the cell where lipase, an enzyme in the lysosome, those little stomachs that contain digestive enzymes, will break down the triglyceride into glycerol and then the three fatty acids. And now we're just going to be considering the picture for even carbon number saturated fatty acids. And so let's look just down from the triglyceride after step 1, and we're looking at an 18 carbon fatty acid on the left side of the figure. Now, we're going to get into something called beta oxidation. And in beta oxidation, we're going to shorten the fatty acid chain by 2 carbons per cycle of beta oxidation. What's the beta part? Well, if we look at that 18 carbon fatty acid, we have the carboxyl carbon. Then the carbon next to it going towards the left is designated the alpha carbon. The next carbon is the beta carbon. And what we're going to do is we're going to cut off a 2 carbon part consisting of the alpha carbon and the carboxyl carbon. And that's going to occur between the alpha carbon and the beta carbon, beta oxidation. And to that little 2 carbon bit, we're going to add coenzyme A. We're going to also add that to the remaining 16 carbon fatty acid. Okay, well now that acetyl-CoA that we made from our little 2-carbon piece can go into the Krebs cycle and will generate reduced electron carriers, NADH and FADH2, which will then send through the electron transport chain. And depending on whose calculation you use, each cycle of beta oxidation will give us between 14 and 17 ATPs. So what we want to get out of this figure and this discussion is that fats are very concentrated sources of energy. And again, there's a reason that we like fats, and that's because they're relatively compact, concentrated sources of energy. Now, we have to go through more effort to utilize them than we do carbohydrates, but the payoff is worth it. So in that caprese salad that I've been talking about, in addition to the polysaccharides from the tomato and the lipids from the cheese and the olive oil, I would have also consumed quite a bit of protein from the cheese. And proteins, we know, tend to be large molecules that are consisting of many amino acids that are held together by peptide bonds. These protein molecules are typically much too large to be taken into cells directly. So as we see in our figure at the top, we have a protein, and the cell will secrete in the extracellular fluid, the environment, digestive enzymes called proteases. And so we will do hydrolysis reactions to break the peptide bonds and turn the proteins into individual amino acids, which can then be imported across the cell membrane into the cell. Now, looking inside the cell, we notice that one of the key steps in processing amino acids is deamination. All amino acids contain an amino group, so we have to deaminate all of them. If we have amino acids that contain sulfur, such as methionine or cysteine, then we will deaminate those as well. Now, our figure shows a 2-carbon molecule with an R group on it, but we'll get rid of that as well. So now we have our 2-carbon molecule that we can modify and react with coenzyme A to give us acetyl-CoA, which can then enter the Krebs cycle by joining to oxaloacetic acid, and then will generate the reduced electron carriers, NADH and FADH2, which we can then use to feed the electron transport chain and generate lots of ATP. Now, there are many genetic disorders that result in the inability to process, to metabolize certain amino acids. One that's common enough that we do widespread screening of newborns is called phenylketonuria, PKU. And people who have PKU cannot metabolize phenylalanine. So chances are, if you work in a neonatal unit, or you may work in one, then you will be conducting the screening. And what we do, because PKU, if untreated, results in mental retardation, we treat the children from a very early age with a diet, a special diet that is low in phenylalanine. What we also do is we counsel the parents of these children to not give them anything that contains the artificial sweetener aspartame. Where do we find aspartame? In artificially sweetened soft drinks, and the ones that contain it will have a warning on the container saying phenylketonuria, that is people with PKU, contains phenylalanine. So PKU is regarded as a treatable cause of mental retardation. We talked earlier about maple syrup urine disease when we were talking about Professor Ames's metabolic tune-up. That was in our discussion of enzyme structure. And maple syrup urine disease results from the inability to process branched amino acids like leucine, isoleucine, and valine. And if one can't metabolize those, what happens is they accumulate in the body and cause brain and respiratory system damage. So we're looking at a catabolism overview. So we start out in our figure with food at the top. Well, we digest food and in the process hydrolyze, degrade macromolecules to form their monomer units. So we'll take proteins and turn them into amino acids. We'll take carbohydrates and turn them into simple sugars, monosaccharides. We'll take fats and turn them into fatty acids and glycerol. What we'll then do is do other modifications of these components so that they can enter into glycolysis, in which case we'll generate pyruvic acid, pyruvate, which we'll take through the intermediate step to make acetyl-CoA, and then take that through the Krebs cycle, also known as the citric acid cycle, and then the reduced electron carriers go into the electron transport chain, and we get ATP at the end. If we're dealing with fatty acids, then we would do the beta oxidation that we talked about, and again generate the acetyl-CoA and continue on through the citric acid cycle, the Krebs cycle, and then through the electron transport chain. In all cases, we're going to generate ATP.